All right. So the problem we're investigating here, guys, is that cells, we already know from the cell theory, carry out the basic functions of an organism. Right? What we want to find out is what structures are going to be necessary for them to do that. Okay? That is the problem that we are investigating. That needs to be in your own words in the form of a question. Okay? So, like, what structures does a cell require to carry out the basic functions of an organism? Something, something. Okay? Now, in this experiment, we will be looking at a plant cell, an animal cell, and a cell from your cheek. Okay? So 2 animal cells and 1 plant cell is what we're going to be looking at. Okay? Which gives us an example of each mode of nutrition. Okay? Or a cell from an organism using each mode of nutrition. So we should be able to get a pretty good idea of what structures would be necessary for any type of cell to carry out the basic functions. Okay? So we are looking at 3 different cells. Right? What we're looking for in each of those 3 different cells is what structures are present. Okay, we want to make a note of anything we see in each one of those cells. We also need to know that all of the cells we are looking at come from eukaryotic organisms. That is, organisms whose cells have compartmentalization. In other words, they have organelles that we can find and look for. Okay? It's kind of important for us to be looking at those cells and not a bacteria where we wouldn't see any of that stuff anyway. Okay? So all of the cells that we are looking at are eukaryotic cells. Okay? All of them are going to be viewed, photographed on high power today. Okay? So there'll be no diagrams done on any magnification other than high.
What we're looking for is just what parts do what kind of thing.
Well, we're not really looking at what parts do what per se, more what parts are common.
Okay.
Yeah. I mean, the what parts do what is important, but more, well, these ones are common, so they must be important. What do they do? Yeah. Okay, so that should help you out with some of the design. You'll notice I didn't outright tell you what everything was this time because I'm kind of weaning you off of that. Okay, so I kind of told you how the experiment is going to go, okay, and accentuated some of the important things, which you could go back and listen to later, possibly would be a good idea. But yeah, to get your design stuff. I did only give you 4 things. You would require for a normal lab report 5, 1 manipulated, 1 responding, and 3 controlled. Okay, so make sure that you have all of that, and that each one is explained separately. Okay, so identify your manipulated, explain. Identify your responding, explain. Okay, the 3 controlled, each one gets explained. Okay, Jack.
Do you have to write a problem in your own words or just a hypothesis?
Problem in your own words. Okay, so for our hypothesis, okay, the if part of the hypothesis should talk about the second point of the cell theory. And after all, that is the problem we are investigating. Cells carry out the basic functions, okay? We're looking to find what structures are necessary for them to do that. So the if part, second point. Of the cell theory. Okay, the and part, as always, describes the experiment. Cells from various eukaryotic organisms are viewed under the microscope and diagrammed. Then, predict what structures you think you'll be able to see in the cells and why. Right? I'm not looking for you to name 15 structures. You're not going to see all of those anyway. Okay, I want you to name the ones you think it'll be possible for you to see and why they would be there. Okay? So the three things we're going to be looking at are a lygostrum cross-section. Lygostrum is a plant, okay, so we're going to be looking at its leaf in cross-section, the thing I showed you actually yesterday, okay. In that slide, this is kind of greatly enlarged, but it'll look a lot like this, okay. This is an enlarged version of what I showed you yesterday, okay. So you can see here, this is the epidermis, okay, up top here, this would be the epidermal cells, okay. These are the cells I want you to focus on. I want you to focus on the cells of the palisade layer, okay, these ones here, right. You will be able to see this level of detail, okay. This is a picture I took that I was looking through the microscope for, so you'll be able to see this, okay. Now, what I'm looking for here is for you to, you might have to use more than one cell to label everything, because if you look right here, like at this cell right here, okay, there's a cell here, but do you see anything inside of it?
No.
So what's probably at the front of that cell that you're seeing?
Water bag.
Yeah, and it's just pushed everything behind it, so it's out of focus and you can't see it. But you could use that cell to label the water vacuole. But you could use the cell next door to label a lot of other things. Okay, everyone kind of follow? Now, when you're labeling, think about this. If I can see the nucleus, and I can see where the nucleus ends, does that mean I can see the nuclear membrane?
Yeah.
It does. Okay? Now, if you also recall, I told you, you should be able to see things like rough ER, but not smooth ER. Okay? And the rough ER, I told you, always look around the nucleus, because that's where it tends to be. It kind of makes the nucleus look like it has a goatee. Okay, or in some cases like a ZZ Top long beard. Okay, so look for that. Okay, you can usually see kind of on the bottom of the nucleus. Okay, I'm going to remove that circle so now you know where to look. Okay, you can kind of see it looks a little bit different at the bottom. If this nucleus was a face, that's why I actually picked this one. Okay, there's like the eyes and the nose, and then there's the beard. Okay, underneath. So. I'm not saying use your imagination when you're labeling, but use your imagination when you're labeling. Okay. Now there are things you can't see. You can't see ribosomes. You can't see smooth ER. Okay. But if you see an open space in the cell, maybe that means you see some cytoplasm. Okay. Or maybe it means you see a water vacuole. Right. As long as you're labeling things and your labels are reasonable, I'm good with that. Okay. If you label the nucleus as the rough ER, I'm gonna go no, that's wrong. Okay, but otherwise you can't really go wrong by putting a label on something unless it's like way, way off. Okay, what do you suppose those green ovals are?
Chloroplast.
Those are chloroplast. Okay, like you can see a lot of detail in the plant cell because the plant cell is really big. I mean, for a cell, really big. Okay, so it's most of the structures are pretty obvious. Okay, so that's what you're going to be looking at for a plant cell. Now on the slide. Okay, what you will see is in the middle of the slide there'll be kind of a, almost like a bow tie kind of a shape. Okay, that is the cross section. So looking at the leaf on this way, right? The best cells for what you need will be located here and here. If you look in the middle, you're going to see all of the vascular tissue, so the big vein that's down the middle of the leaf. You're not going to see the cells you want to see there. You're going to see hollow tubes, okay? And that's not the part of the leaf we want to be taking a picture of, okay? So kind of halfway down either side of the leaf from the middle, okay, is the best place to look for those cells on the slide, okay? Now, when you look at the animal cell, okay, and that's what it's labeled, animal cell, it's actually stratified squamous epithelium. Okay, which is the layer of skin underneath your epidermis. So if you dust your knuckle, right, and you peel off that skin, and then there's that skin that bleeds like crazy and stings like crazy right underneath, the red stuff, that's stratified squamous epithelia. Okay, that's what you're looking at, right, in this slide. So it's full of animal cells, and it looks like this. Okay, or this, depending on which stain you get.
What,
All right,
what critter is it from? You know?
probably a person.
Probably a person.
Yeah, from a cadaver.
No.
Yeah.
No, I just put some skin on civilian and called it a day.
Ew.
Well, technically,
No.
well, technically, just a dead person.
Yeah, but I mean, people donate their bodies to science, so that's, yeah, if they're used for like gross anatomy or, you know, study and things like that. Okay, so in the animal cell, okay, this is what you're gonna see, okay, depending on which one you get. The darker stained one is sometimes easier to see, but this is blown up a bit from what you will see. Okay, so it kind of looks like, you know, when mud dries, it cracks. Okay, it kind of looks like that. That's the texture you're kind of looking for. Okay, but the cells are quite a bit smaller. So just so you're aware, each one of these circles would be a what? The nucleus. Each one of those circles is a nucleus. Okay, so once you find one, you can kind of figure out where the rest of the cell is. So like here, that's the rest of that cell. Okay, and I can see some structures in there. You're probably not going to see as many on the animal cell as you saw in the plant cell, and that's okay. Okay, do your best to label what you can see. Right? If I'm looking at this a little bit bigger, okay, so any one of these here is a cell. Okay, look at all the nuclei that are in there. Okay, pick a good one, do your labeling, or use more than one to do your labeling, whatever you got to do.
Are we guessing the size of them?
Yes, of a single cell. Okay, because you're diagramming an animal cell. You can't say, well, the sample took up the whole field of view, so it was 400 micron. No, okay, you got to tell me how big was that individual cell along the longest dimension.
So each of the pages we're doing it the exact same as we did last one,
Yes.
just we're labeling actual things.
You're labeling accurately this time, yes.
And how many labels are expected?
As many as you can be reasonably certain of.
We're also expected to measure each one again?
That's what we just asked.
Okay.
Yeah. Okay, now for the cheek cell, you're going to be making a wet mount of your own cheek cells. This one's mine. I took this picture. It's my own cell, and I'm quite proud of it
Yeah.
because it's awesome.
Was it alive at the time?
Yes,
Yeah.
you will be looking at those cells alive.
Will we be able to see them move?
Yeah,
They don't
that's
move much, no,
around.
no, because they're sloughed off, so they're already kind of, like, you'll actually see cells that have been torn apart by the process. Like, you'll see debris and stuff like that. You'll also see, regardless of how well you brushed your teeth or washed your mouth out, okay, this morning, there's still gonna be food debris if you had breakfast, especially if you had something fibrous. And if you had like a whole grain toast or something like that, those fibers, they catch in your mouth, they stay in your mouth. If you see something big and dark, it's not a cell. The cells are actually quite transparent because we're not staining this. Okay? All of the other ones have had a stain added to them, so color has been added. These will not have that, so they're a little bit tougher to see. Now, in order to help you with that, once you think you have something on the high power, on the bottom of the microscope, so underneath the stage, okay, there's a condenser diaphragm. Okay, so there's this kind of structure underneath the stage, and there's a little tab on it. And if you move that tab, it closes off an iris inside. If you close that off a bit, it actually increases the contrast of what you're looking at. So it accentuates the difference between light and dark on the image, and it can help that cell to pop. Okay? When I first found this cell, I barely noticed it was there, and then I closed that iris off a little bit and it just popped right out. Okay, and it was quite obvious to see. This one here, I closed it off a little too much, and you can see that it got grainy because of how dark I ended up making it, so the picture wasn't as good. Okay, so that's a single cheek cell. You can see quite a bit of it. Okay, quite a bit of structures in there. Do your best to label. Okay, here's the process for acquiring your cheek cell. There will be a box of Q-tips on the front table. Okay, please, in the interest of, germs, pick them up by the middle. Okay, try not to touch the swabby end of any of these because other people are going to have to put this in their mouth. Okay, so to get your cheek cells, okay, you just put it in your mouth and twist it around and around between your fingers a little bit, kind of like what I'm doing here. So
It's like COVID test?
Uh-huh. Wow, COVID test goes up your nose.
Yeah.
Don't stick this up your nose, okay?
Would that have cool cells?
No, it would have a lot of mucus. Okay, so, and now when I put it on there, I do the same thing. I'm still twisting it, but I'm smearing it back and forth as I do so. Okay, just like that, couple of times. Drop a water cover slip, put it under the microscope. You've got cells on there. Okay, that's why they can do DNA tests like in CSI, just by swabbing the inside of somebody's mouth. Okay, it's exactly the same thing. Lots of cells will come off because the inside of your mouth sloughs cells really, really easily. Okay, when that's done, that goes in the garbage. Okay, the cover slip goes in the garbage. Wash and dry with soap. The slide, because it has, after all, had, well, your mouth on it. Okay, so make sure you wash and dry. Okay, wash with soap and dry before returning it to the front. Okay, everybody with me there? Okay, we'll talk about the analysis when we, actually no, we'll do it right now just because we're here and we can do it. Okay, so the analysis, there's only one analysis question if anybody looked. Okay, based on your knowledge of cell structure and your observations of the cells in this lab, what organelles are most important for a cell to function and which ones are specific to a certain mode of nutrition? Explain your reasoning. So, I'm looking for a paragraph here. Okay, in that paragraph you tell me what structures you saw that were common. You tell me why they were common, why you saw them, what, maybe a little bit about what job they do. Especially anything related to the two modes of nutrition that we talked about. Would it be a good idea to include those two modes of nutrition in your answer?
Yes.
Yep, I would. I'm going to be looking for that, so make sure you do. Okay, that's what I'm looking for. Paragraph answer that does that. Okay, that's your analysis for this lab. Conclusion, same as always. Copy paste your hypothesis and then talk about your data. Did you see the structures you predicted? Why or why not can you accept or reject your hypothesis? Sources of error. Okay, not inexperienced this time. You're now experienced. So think about maybe some other things, okay, that could cause you a little bit of difficulty, okay, in this lab. All right, questions? Okay, the biology lab is already open. Okay, so very quietly make your way to the biology lab and grab a microscope and we'll get started.
Should we sign out and shut these down?
Yeah, sign out and shut down, but just, just in case. Leave it on your desk. If we get a little bit of time, we come back, we'll use them.